from our Colorifix, one of our sponsors. So Colorifix is the first company to use an entirely biological process to produce, deposit, and fix pigments onto textiles, inspired by nature and engineered through synthetic biology. Their dyeing technology uses zero harmful chemicals while significantly reducing water, energy, and waste. So Colorifix is working with global dye houses and fashion brands to transform industrial dyeing into an environmentally friendly, socially responsible, and financially viable process. So before I introduce our speaker today, I'd also like to just share a bit about our upcoming events. So next week, we actually have Dr. Arvid in person talking about the gene's eye view of evolution next Thursday. And we also have an upcoming pizza social where you can come and get free pizza, drinks, and meet other people, which is happening on the 16th of February. We'll send out more details as the date comes closer. Yep. So today we're really honored to have Prof. Dame Caroline Dean as our speaker. So she's currently a Royal Society professor based at the John Innes Center at Norwich, and she's also a fellow at the MRC Laboratory of Molecular Biology in Cambridge. And she has won, amongst many other things, the Genetic Society Medal in 2007, the Darwin Medal in 2016, the L'Oreal UNESCO Award for Women in Science Award for 2018, the Wolf Prize in Agriculture 2020, and the Royal Society Royal Medal in 2020. So without further ado, let's welcome Prof. Dean to give a talk.
Well, thank you very much. It's a pleasure to be here. Sorry I'm not actually there, but what I'm going to do today is give you an overview of what my lab's been working on for many years now. And that's this question of how plants know what season it is and how they align their development to the seasonal cues. Now, many organisms use environmental cues to affect their gene expression, but plants are really masters of this. They use daily seasonal cues to time their metabolism, their development, and their whole growth pattern. And so as I go through, I'm going to tell you what we've been doing, hopefully at a level you'll understand. I will stop every once in a while and ask, are there any questions? And so please ask me a question in the middle of the talk if I'm pitching it at too high a level, because it'd be much better to have a conversation about something you understand rather than me drone on about something that's a bit too complicated. Anyway, so we started off thinking about this question many years ago when I started my lab, just how do plants figure which season they're in? And this picture of this tree growing through different seasons really beautifully exemplifies that. Now, if you think about the seasonal cues you know about, you'd probably say, well, day length is one and temperature would be the other. Those are the two major cues in this part of the world, at least. And day length, of course, is the same in autumn and spring, so that the major cue used to time spring after the period of prolonged cold is temperature. And that's what we got interested in. How do plants know they've overwintered and now it's time to, in our case, flower in spring? Now you might think, okay, well, it's just a question of monitoring long-term cold exposure. But the graph on the right shows you actually the temperature profile the plants are actually seeing. These are plants in a field in Sweden with the temperature loggers next door to them. And what was striking really was just the extent of the variation of the temperature on a daily basis, more over a 24-hour period than over, in fact, a season. So the plants, to monitor the seasonal progression, have got to somehow remember past memory of cold exposure as well as the current temperature. And they also have to cope with this very noisy temperature. How do they extract a signal, a long-term signal, from a profile such as this? And that was the question we set ourselves. Now, 30 years ago, we started this. But of course, at the time, there was very little understood about the molecular basis of any of this. We knew it was for the question I picked, which was the seasonal control of flowering that was affected by these long-term cold exposures. That was a process called vernalization. Very important for many native plants, but also for a lot of crops. A lot of genetics have been done in breeding for winter and spring sown varieties of wheat and brassica and many of our other major crops. So the winter types would germinate in autumn and overwinter, and the spring types would be sown in the spring. And this is actually what Arabidopsis looks like in that scenario. You have winter types and spring types of Arabidopsis, which if you give them weeks of cold, just as you've germinated them, this extended period of cold slowly accelerates the flowering. Now, what was interesting about vernalization, and this was true for all of the different plant species, was that this temperature exposure was quantitative. The more cold you had, the earlier the flowering. So it was a quantitative response somehow. It was not graft transmissible, but it was mitotically stable. And that if you took a cutting from a plant that had had this cold exposure, all of the cells remembered they'd had that cold exposure. It was mitotically stable, epigenetic. And then the regenerated seedling and the regenerated plant would not have to go through winter again. And that was what intrigued me, as what the memory of this cold exposure was, this mitotic memory. Now, the hypothesis at the beginning of the work was that there must be some floral specific regulator that was changing in response to cold. And in fact, there were actually mathematical models that the breeders and the people growing crops used. And they would measure temperature over days and measure it within an interval of 1 to 10. And there would be a model to predict that, you know, with that particular temperature exposure, the plants would flower at X date. And so that was our starting point for trying to understand what was going on. Now, just to set the scene, of course, temperature is not the only thing that affects flowering. And here's a sort of schematic to give you an overview of actually, of course, many things affect flowering in many plants. And different plants all need different physiological cues. And when we first started, we did everything in Arabidopsis, so we could do a genetic analysis. And one of the first sort of realizations was through this genetic analysis and looking at the different inputs was that what was apparently such a physiological complex thing, in that different plant species all needed different inputs, could be simply explained by the fact that all these inputs converge to regulate a common set of genes. And in fact, as we've gone in to do the molecular mechanism, we can now go back out to other plants and say, well, okay, these plants don't need cold, they need photoperiod. But by understanding the mechanisms, we can then really understand the evolution of flowering time in many different plant species now. And so we published this rather provocative review about Arabidopsis as the Rosetta Stone of flowering, but it's still actually, the conclusions in there relevant for flowering generally. But it was winter I was particularly interested in. And again, you know, we used Arabidopsis. And in fact, most people, if you've ever seen Arabidopsis, you would imagine in your mind this plant on the right, this rapid cycling type. And in fact, a lot of Arabidopsis are rapid cycling. The ones people use in genetics generally are. But in fact, most Arabidopsis, if you pick them from outside here or further north and put them in your growth room, they look like this. They keep making leaves, and they make a lot of leaves before they flower, unless you give them cold at the germinating seed stage. They have this requirement for cold, for prolonged cold exposure, to flower at this early time. So the idea then is that some plants have this winter annual habit, and they're adapted to overwinter and flower in spring, whereas others are adapted to this rapid cycling or summer habit, and they germinate in early spring, and they cycle much more rapidly. They don't need winter. And the advantage of this in some climates is that they can get in 2 generations a year. Whereas, of course, this strategy means it's a safer strategy. You avoid summer drought, but you are limited to 1 generation. And there's a lot of ecology and a lot of interesting evolutionary discussion about reproductive strategy, and it's all based on to overwinter or not is a large part of that. So we had this genetic variation in Arabidopsis, so we could go in and ask genetically what causes some varieties to overwinter and others not, so we could understand the molecular basis of this decision. For those that needed to overwinter, how did they register these complicating fluctuating temperatures over many weeks and months? And then the third question, which I won't have time to really get into the answer, is, and winter, of course, is different in different parts of the world. So how does Arabidopsis, which has a very big geographical range, pretty much the equator up to the Arctic Circle, how does Arabidopsis adapt to different winters? And we did a classic forward genetic approach, mutagenesis, natural variation. We started this a long time ago. And I think if we did it now, we'd probably do it with an omics approach, and we'd end up getting very bogged down in a lot of detail. And I think the fact we went with the forward genetics really simplified our conclusions, because all our genetic pathways took us to the study of one gene. It's a gene that expresses, it's called FLC, it's a gene that expresses a protein. It's a transcriptional repressor, Madsbox repressor, that keeps off all the genes needed for the meristem to switch to floral fate. So it's like a brake to flowering. If the protein is at high level, plant stays vegetative. So this is my attempt to schematically represent what you've just seen. So if FLC is expressed at high level, the plants stay vegetative. This is then what determines whether or not they need to overwinter. And as I'll tell you, the prolonged cold is then an epigenetic silencing of this same gene to allow the plants to flower in spring. So all our questions are questions about whether or not the plants needed to overwinter. It came down to a quantitative transcriptional analysis of this single gene, and whether or not or how the plants registered cold came down to actually a very conserved polycomb-mediated epigenetic switching mechanism. And so we've ended up really sort of starting from a plant question, getting into the heart of chromatin regulation. And as I say, the natural variation, which I won't have time to get into, is really modulation of either this quantitative transcriptional regulation or this switching, epigenetic switching mechanism. Okay, so I'm going to show you quite a lot of schematics about FLC expression through the life cycle. So as the plants germinate in autumn, if they're a winter annual type, they start off with high levels of FLC transcription's high. Through the winter, FLC then is slowly switched off. Now, as I showed you earlier, there's a progressive acceleration of flowering. Well, that's because of a progressive silencing of this repressor. So it's slow and it's quantitative, and I'll try and explain to you why that is. Now, the spring types actually do not express very high levels of FLC. So they don't need to overwinter. They already have low FLC. So that's how quantitative transcriptional regulation affects this reproductive strategy choice. And then the quantitative silencing is the registration of this complex temperature profile. And then as the plants come out in the spring, FLC is then maintained in this very low level through the spring and the summer as the plants flower. And then only when they flower, and this is a seed pod of Arabidopsis, one of these little seed pods here, does FLC expression get reset so that every generation, when it germinates, is a winter annual, if it starts as a winter annual. So in fact, what I'm going to show you is this, how you have a quantitative transcriptional regulation, how it's silenced, and then in fact, it's how it's reset. And all our questions that we kept coming back to, looking at this, took us back to a chromatin mechanism involving histone modifications. And I'm going to explain those a little bit, but if you're not into histone modifications, all you need to understand is a sort of color coding traffic light system. If they're green modifications, the gene is active. If they're red modifications, the gene is silent. Now, just to start, I'm going to go explain that what I'm going to do is start, if you like, from the end. I'm going to explain this long-term memory. And then I'm going to go back in the talk and talk about the switching mechanism. And then lastly, about the transcriptional shutdown. And I'll come back and layer onto this schematic. So we got into this chromatin mechanism through mutants. And in fact, classic forward genetics took a plant that needed vernalization. Normally, when you vernalize, they flower early. But after mutagenesis, we picked the ones that flowered late. And this took us into this chromatin mechanism involving polycomb repressive complex 2. Now, it's very well conserved. It's important in mammals and Drosophila. It's most well characterized in Drosophila. So we can really go in and use a lot of the biology that's well known. But we also got out of our genetics 2 other proteins, which I'm going to call PRC2 accessory proteins. This one, you have to remember this name. There's 2 names you have to remember. FLC is the gene that's silenced, and VIN3 is the cold regulated accessory protein. And those I'll talk about a lot. So just to make sure everybody's with me, polycomb silencing influences this epigenetic regulation through histone modification. Now, if you unravel a chromosome, you get basically the chromatin. The DNA is wrapped around these histone octamers. This is my attempt to draw those histone octamers, and the histone tails are modified chemically. We just have to worry about a K36 trimethylation, which I've color-coded green here, or a K27 trimethylation of color-coded red. And PRC2 is always delivering this red modification, K27 trimethylation, associated with silenced chromatin. And, this is the sort of schematic, which is as simple as I can make vernalization. And if you can understand this, then hopefully you'll understand the rest. So the gene is on in the autumn. You have the modifications green, RNA's pumped out, proteins made, the break is on. None of the other genes that needed to switch to flowering are made. The plant stays vegetative. Slowly but surely, during the winter, there's a switch of these histone marks now to red, K27 trimethylation, and that happens in the cold. When the plants come back into the spring now, instead of the gene coming back on, they are epigenetically maintained in that silent state. These red marks are inherited through many cell divisions in epigenetic inheritance. And so the FLC gene stays off. And that really is this epigenetic inheritance that I want to make sure you understand. There's been a lot of stuff in the literature about, transgenerational inheritance, and that's really what's caught the public imagination. In fact, very few epigenetic mechanisms are transgenerational. Most things epigenetic are about the mitotic inheritance in the organism of that generation, and that's true for FLC. Okay, so K27 trimethylation you can measure using a chromatin IP experiment. You can buy an antibody that picks up that modification, make chromatin from plants and do an immunoprecipitation, and then track along the gene of interest. And we're looking here at the bottom of our gene FLC. And with lots of primers, you can then measure the K27 trimethylation in plants now that have been exposed to different weeks of cold, 2, 4, 6, 8 weeks, and then you bring them out and you grow them for 7 days. So this is the stable, silent state. And what was clear was that the more cold you have, the more K27 trimethylation you have. Now, when you're a molecular biologist, you think, oh, well, that explains, here's a nice quantitative decrease in my gene expression, and it's associated with a quantitative accumulation of this silencing mark, so that there's quantitative regulation at the locus. In fact, that's the wrong conclusion. And what I'm going to show you now is that this is actually an average of a whole set of cells, which are either K27 trimethylation or the green mark, K36. And it's a population average where you switch from one state to the other. But because we grind lots of cells together, it looks like an average like this. And as molecular biologists, we endlessly make this mistake. So it's something to think about. So what we have, I've got the schematic on the left and the real plant on the right here. So I'm showing you roots because actually we can image roots very beautifully, but the same is true in the rest of the plant. And the nice thing with roots is in Arabidopsis, they grow from the meristem down here. And the cell division is all in one direction. It's called anticlinal, it's horizontal. So that all of the cells in these columns are actually daughters and granddaughters of something down here. It's a developmental lineage. And so if all of the cells are on, color-coded green, as the plants germinate, and then they're exposed to cold, what happens is that there's a switch in one of the cells from green to red, and then all the daughters, the mitotic daughters of that cell, stay red. But as the plant is experiencing more and more cold, more switching happens. And then as you grow out, and this is the growth back in the warm, following transfer back up in the spring, more and more of the cells then become switched off until the end, all of them are switched off. But of course, if you grind these up, you get a nice quantitative change. So if you look on the right, this is actually a fusion of FLC with Venus, so we can look at it in the nucleus. The roots on the left here, they're all on. but looking over here, let's say the 8 weeks cold, you can see there's a beautiful lineage of cells where the FLC is still on, whereas the next door, it's off. So it's a cell autonomous switching mechanism where there's memory all the way through the daughters and all the way through to the seed resetting. So this quantitative silencing is just now a slow switch. And once you've switched, which is why we call it digital, it's an all or nothing. That is then memorized for the rest of development. Now, flowering is not sectored like this, and that's explained by the fact a downstream component, a target of this protein FLC, is a gene called FT, which makes a little protein that moves around the plant. So in fact, the sectoring of FLC is then averaged by protein movement in the downstream step. Now, if anyone wants to shout or send me a chat or anything like that to say, this is ask a question, please do. But what I've showed you so far, if we look at our schematic, is that when the gene is on, and I've just represented the gene by a series of nucleosomes with their green mark, and we've got transcription with this line here, the gene is on in autumn. And as what I've shown you is that it's by the time you're in spring and summer, it's off with all of these red marks. And it's a digital switch. It's either on or it's off. In fact, it's then reset in the seed, and I'm not really going to have time to talk about that, because what I want to focus on is this sort of winter period, because it's such an interesting story, and of course so important as our climate begins to change. So now I'm going to work backwards into this cold part. So what happens to the K27 trimethylation actually during the cold? So what I showed you before was on the bottom here. You see it spread over the body of the gene. In the cold, what you see is accumulation of this modification, mainly in one part of the gene. And if you track back at the bottom here, that's just downstream of this transcription start site. Now, we call this the nucleation region. It's not in the promoter, interestingly, but it's a really important switching mechanism where actually what's in the cold is a localized switch, which then spreads across the body of the gene only upon transfer to the warm. Okay, so what is this nucleation region? Well, this is why the genetics was always so good to us. We kept going back to the genetics. The genetics identified a mutation, very strong loss of vernalization response mutant, which was a single mutation actually in the intron of FLC. Now, of course, when you normally search for mutations in your mutant collections, you're looking for things that disrupt the open reading frame. So, you often overlook these intronic mutations. But this one turned out to affect a cis element in the beginning of the intron, in the nucleation region, which affected the binding of a protein, which is a DNA binding protein. It doesn't really matter what its name is. It's not just specific for vernalization. It affects many proteins in the embryo, many gene expression in the embryo. But through a sort of proteomic walk, we took this DNA binding protein and identified partners. And we get to now these PRC2 accessory proteins that I'll talk about, which had come out of our genetics right at the beginning, and our PRC2 complex. So we'd managed to link the proteins that we knew were important from genetics to this nucleation site. Okay, so nucleation then is a complex, multivalent association of proteins at this localized site. And I've drawn it here as 3 nucleosomes. It's quite a limited site just downstream of the start site of transcription. And Val1 is the DNA binding protein that pulls all of these proteins together. Now, why is it so slow? Remember, this takes weeks and weeks for all the cells to switch to the silent state in the meristem and then have all the daughters be silent subsequently. Well, part of why it's so slow is that one of these accessory proteins, VIN3, is induced very slowly indeed, maximally by about 6 weeks of cold. So let's just have a think about what happens at that nucleation region and why it's so cold. So I'm going to show you why it's so slow. I'm going to show you now the increase in K27 trimethylation and the decrease in K36, because this is quite cool. So as one goes up with increasing cold, the other comes down. So what we have is this slow accumulation of this complex delivering K27 trimethylation. It looks quantitative when you do the grinding up, but it's an on-off switch where you go from green to red. So in the cold then, it's at the meristem, the cells are switching slowly, the meristematic cells, and here I've drawn 2 switched, 3 switched, and so on. That is very slow because of one of these accessory proteins that's required for that switch, which is slowly induced by cold. And then you come back into the warm, and if you're switched, you grow rapidly and all the daughters stay off. And then it's the proportion of cells that are off is giving you that quantitative silencing. So I hope that's clear. That's this digital silencing I make a case about, because I think in the end, what we will find is most chromatin memory mechanisms are digital. And that's not really a sort of well-known fact, I would say, in the literature at the moment. But I think as we've got into modeling and really understanding the polycomb switching, I think to have a memory state to be able to be inherited, there has to be a digital state, which is then switched to the daughter. So that's one really important thing we learned from FLC. Okay, so why is it so slow? As I said, there's this accessory protein, VIN3, which is very slowly upregulated. And when we really wanted to understand, I wanted to say it's slowly regulated, but somehow it's got to read these complex temperature profiles, the complex noisy temperature. How is it doing that? Now, this set us into a project where we actually did a lot of field experiments. I was very fortunate to get this funded because, I'd never done a field experiment in my life before, but it seemed to me that we needed to go out in the field and understand what was going on in a real winter environment, as opposed to our cold rooms where we just put the plants for 2, 4, or 6 weeks in nice constant temperatures. So we were helped enormously by people who live in and work in Sweden, by Svante Holm, who works at Mid Sweden University, which is right up in the north here, and by Torbjörn Sol, who works at Lund University. And we had 2 field sites in the North Sweden, right up with the Arctic Circle, and a master's student, Christina Berggren, we sowed in September, October. And then every week they would go out to the field, dig the snow back, harvest the material, and send it to Norwich so that we could understand the upregulation of VIN3 if it had happened, and the downregulation of FLC in our normal natural winter. And we'd also set up a greenhouse in Norwich where we could we'd sort of taken all the lights out and all of the heating, so we could include some transgenic lines. So this was our sort of cannibalized greenhouse, which substituted as a field experiment. It was a really great couple of years. We did 2 years. We harvested and monitored all the way through the winter. So I can only show you a little snapshot of some of that data, but what was interesting was we took the temperature profiles in the different places, and that's what's shown at the top. So you can see very different winter exposures. We had 2 plantings in Sweden because we weren't quite sure what was going on, but fortunately they really looked rather similar. And then we harvested the material, we, being the master student, sent them back quite regularly over the whole winter period. And then we measured this VIN3, this cold-induced accessory protein. And also we looked at the decrease in FLC RNA. And, one thing was reassuring was that in the field, we got FLC went down and VIN3 went up. We weren't absolutely sure that chamber experiments would completely recreate the field, but they did. But what was interesting was when you looked at the differences, that overall they were rather similar. But actually in North Sweden, there was a nice exponential decay of FLC expression and a nice fairly linear increase of VIN3. But actually in the Norwich, well, this was the data, and this was the exponential plot, the log plot, didn't really fit very well. And actually VIN3 took an awfully long time before it started to increase. This was a really warm autumn that we'd had in Norwich. Now, when we mined in, and I'm going to show you this one in bigger detail now, that's this same plot. When we looked hard, we thought actually there may be 2 phases of FLC silencing here. And that made sense when we looked at the mutants that we collected, because this mutant that now didn't have accumulation of this accessory protein kept silencing at this slow rate. And there was a switch which coincided with when VIN3 was upregulated in the field to a much faster FLC silencing. So that was nice. There was this two-phase silencing depending on VIN3 accumulation. But we clearly needed to know about how these noisy temperatures were affecting VIN3 accumulation. Now, we were fortunate to be able to have chambers in Norwich where we could recreate exactly what was happening in the field. So we had a northern Sweden winter, and that's the orange here, where every 24-hour period we gave this was the southern one, a 14-degree mean, but in a step. So you went down and up every 24 hours with a mean of 14. And then we compared that with a chamber which was constant 14 degrees. And then we had the northern Swedish type, which was a mean of 8, but again, a step change which went low, 6 degrees or so, up to about 11. And we compared that with a chamber where we had a constant 8. And then we had plants with this every 24 hours, and we monitored over days what VIN3 was doing, the accumulation profile. What was surprising was that the constant and fluctuating 8 really was no different, but there was a big difference between the 14 fluctuating and constant. And I go through this because actually these kind of experiments were so revealing that I think we've got to go back and do this kind of experiment for much more of our thermosensing analysis in plant biology. Because what this told us was that the VIN3 induction, far from being reliant on cold, which everybody had assumed it was ever since it was discovered, relied on the absence of the warm temperature. It was the warm temperatures in the afternoon, these peaks, which prevented VIN3 accumulation. Now, the other thing we did, and I'm not going to go through this, but it was published a couple of years ago, was do a screen to look for regulators of VIN3, cold regulators. And that took us to a protein which was a direct regulator, but it was not itself affected by cold, but it was a very stable protein. And so what happened in the cold, and again, I'm showing you roots because they're easy to image, is because it was a stable protein and a very slow accumulation just in the meristem, the protein accumulated because of reduced dilution due to slower cell division in the cold. And it was this slow cell division which gave us this rather slow and long-term accumulation of VIN3. Because if you think about it, most things that are temperature sensitive, responding to the immediate temperature, it was actually quite hard to think of something that would be then slow and so long-term. And so it's an indirect cold upregulation through a growth effect. So indirect, and that was really a big surprise. So what we have now is the plants are actually monitoring these fluctuations, both the long-term and the acute fluctuations for VIN3. It's accumulating slowly over a very broad temperature range, but it's very slow through this stability of this protein that was an immediate upregulator. But as soon as the temperatures went too high in the afternoon, anything that accumulated was turned over very rapidly. So you needed to have the absence of autumn warm temperatures before you got very high accumulation of this accessory protein. And that these fluctuations in long-term accumulation of the cold was then locked into place by this switch to this nucleation of polycomb silencing. Now, we went into this a little bit more. There was a paper where we got into modeling, where we could show this long-term, the short-term that I've told you about, but also a current temperature that actually, as well as the accumulation of the protein, the actual nucleation activity was temperature dependent. All of these things input into both accumulation of VIN3 and the nucleation of this polycomb complex at FLC. So we have now not just one or two major thermosensors, but distributed thermosensing through the whole network. And again, that was totally unexpected and really has changed the way we sort of think about how plants are monitoring their natural environment. And of course, this has got huge importance for predicting plant behavior as the climate changes. Now, I'm just going to finish the talk by talking about this VIN3 independence. So far, all the cold I've told you about is all about VIN3. But in fact, there's another major cold step, apart from the fact that the cold is affecting the whole Vin3 network. That cold also affects this earlier step in the process before the nucleation happens, early in the cold exposure, and that involves non-coding transcripts at our gene. In fact, in our case, antisense transcripts. Now, again, this is a big area in chromatin biology. The role of RNAs in modulating chromatin states is fairly controversial. There's lots of papers. And so this system has actually proven to be quite useful to unpick just what's happening at one gene in an environmentally switchable system. And it turns out that as the plants begin to feel the cold, then you get much stronger expression of the antisense transcript. This is the schematic. Here's our sense transcript that I've shown you before. You get the antisense transcript starting at the sense polyase site and running all the way through to the sense start site. They cover the entire transcription unit. Now, they're very complicated. There's lots of our papers, but I just don't want to complicate it too much. It's just that cold significantly upregulates the antisense. That's a transient pulse of antisense, okay? And when we looked at, because of course we wanted to know, is it a switchable manner? When we looked in the nucleus using single molecule FISH to look at the molecules of RNA, green shows you the antisense transcription in the nucleus and red shows you the sense with the different flaws on the different oligos. What we found was that you only ever at each locus saw red or green. You never saw both together or yellow. And that showed us that we had mutually exclusive transcription of these two strands. And so if the antisense goes up, then the sense goes down. Now, we're still working out exactly how that happens. But one interesting thing that came out of the field that helped us elaborate that was that in one of our field experiments, when we were looking at the expression of these antisense transcripts, which were christened couleur, for a long story, you can ask me afterwards. What we found is on one day, there was this massive accumulation of couleur in many of the genotypes that we were looking at. And the temperature on that day, if you look in the box up here, that was the first night the temperatures dipped just below 0. Now, in our chambers, when we recreated that, and we did that by fluctuating temperatures, giving all the plants the same, we had 2 weeks of 5 degrees, either as a constant 5 degrees, a mild fluctuation that didn't get to freezing, or a severe fluctuation that did get to freezing. What we saw was that this only occurred upon freezing. And when that occurred, we saw a very strong reduction of our FLC RNA. So really, as the plants were perceiving that first freezing temperature, they switched off the sense transcript. And one way they do that is that the antisense sequesters in what's now, again, another interesting area through a condensation mechanism, which you can see in these puncta. This is Arabidopsis nucleus, and these puncta are condensates with an important transcriptional activator of FLC sequestered away from the locus by this antisense transcript. And if you're interested in this, that just came out recently. So we can actually make this figure even more complicated now in terms of how the plants are registering these complex temperatures. You've got in the warm, strong activation of transcription through this protein called frigida, which is the sequestered protein. As soon as it's cold, that protein is taken away from the locus, sequestered by the antisense RNA into these condensates. That as soon as you have a cold night, you have suppression through a different mechanism on the transcription of FLC. And of course, those cold temperatures are also now inducing this Vin3 accessory protein. And as soon as you've gone past the warm autumns, then this begins to accumulate. So now you have shutdown of transcription aligned with now nucleation of your polycomb silencing, which is much more slow, and means that you need the whole of winter before actually you have the memory of the winter. If you come, this will work, and if you bring the plants back out again, transcription will go back on. So this is a transcriptional plasticity. This is now the memory of the winter exposure, the nucleation. So I hope what I've managed to do is convey to you in a somewhat simple way how we've got an ability of the plant to register these complex temperatures through these antisense transcription dynamics affecting the sense transcription, that we've got acute and long-term inputs of the temperature. In addition, we've got this much slower induction of VIN3, which causes then, once the transcription's down, the switch. And once that switch happens, the plant is, it doesn't really matter what the temperature is doing. That is epigenetically silent and stable all the way through the rest of development until the seed. I haven't really gone into this, but as the plants come back into the warm, there has to be a spreading over the whole locus for long-term memory. And then everything is reset in the seed to allow the expression to come back on in the right way at the beginning of the next generation. So we've got really conserved co-transcriptional mechanisms here. This is all about transcription. This is, none of this is plant specific. None of this is in cold specific. There's a lot of developmental and environmental inputs that could substitute for the cold here. So we have transcriptional shutdown, an epigenetic switch involving this very conserved polycomb silencing mechanism. And we've managed to now unpick the mechanism of how it really works in a sort of real natural winter. So the two messages I want to leave you with are, chromatin and polycomb silencing, which are widespread in biology. I think we need to think of them as digital states where you have opposing states. Remember our traffic light system, green and red. When we do omics, we of course average everything, but I think if you look at the cellular level, we'll find that that's mainly a digital state. And that digitalness allows a memory state. And I haven't really got into that. That's what we're doing at the moment in collaboration with people at LNB, how these accessory proteins, once they're there, confer memory through mitosis. And the second big kind of thing, which was a surprise, was this temperature sensing mechanism. That the idea of one or two major thermosensors, remember the crop modeling I explained at the beginning, was not really the way it's happening. That temperature is affecting many, many aspects. I mean, temperature affects many aspects of biochemistry. That's what's happening. But there are nodes within the network then that integrate those different temperature inputs. So the temperature dependence is distributed through the network, but there are nodes which capture it, and the nucleated state is then the full capture, if you like, of the whole winter fluctuation. And this kind of thinking, as we go forward with the changing climate, I think is going to be very important. It's not just true for Arabidopsis, it'll be true for most of our crops. And this is why I went into the Rosetta Stone thinking, we can translate this out much more broadly. Okay, I'm going to hopefully have lots of discussion with you now. I just want to thank a lot of people. This is work that's been going on a long time. With a lot of funders, but it's a big collaboration with a colleague of mine at John Innes called Martin Howard, who's actually a theoretical physicist by training and is a computational modeler, who's really got me into thinking about this digital states. And a lot of the work, which I haven't really got into, he's gone into modeling these epigenetic states quantitatively. And that's been a really very insightful approach. And the other is that I want to acknowledge the people at LNB, Marianne Biensis' lab, and Elsa Franco-Echevarria, who's the postdoc who is in Marianne's lab, and Mark Fiedler, and they're working away on this mitotic inheritance mechanism. The people in the lab, this was a photograph a long time ago, because of course we're all standing next to each other. This is pre-COVID. The people in yellow are the LNB people, the people in red are the computational biology people, the people in white are the ones who are still in the lab, and we have some new arrivals. And it's a great team effort. We have some fantastic conversations, and I acknowledge that they've worked so hard to allow me to give these talks. And let me stop sharing and hopefully take some questions from you all.
So thanks, Prof. Dean, for giving the talk. Does anybody have any questions they'd like to ask? So you can feel free to type it in the chat, or if you're more brave, you can unmute yourself and ask the question.
Please do unmute, and you can ask me any question, nothing to do with the seminar. How on earth did you end up picking vernalization as a topic, or anything? Let's just have a nice discussion.
So hi, first of everything, thank you for the talk because it was very, very interesting and stimulating. So something that I wanted to ask you is, have you got into considering the light sensors in all these circuits and how they integrate into the winter sensing?
Yes.
Many plants also.
Absolutely. No, that's what I had at the beginning. You know, the day length is an important factor in all of this. As far as we can see, they're rather independent. And I would say the light sensing for most of the overwintering types comes into play in the spring. So basically, for the very strong winter annuals with very high levels of FLC, they're not very day length sensitive. So, you know, FLC masks everything. Once FLC has gone away by the winter, now the light sensing becomes very important. That downstream target I was telling you about, FT, is very photoperiodically sensitive. So not only do you release the repression mechanism, but now the day length promotes FT expression and other things. And the integration then happens at nodes like FT expression. So there's a lot of understanding on that. It's just that we've focused in on the temperature.
And something else that I wanted to ask you is, so I said you get directly into saying that you went into the root and into the germ of the root to see how this was influencing the temperature. How did you know that it was there? The sensing center was in the root and not in some other parts of the plant?
No, I misspoke if that's what I said. Basically, all the cells sense the temperature.
Oh, okay. Okay.
If you look, you know, it's just the things we followed. FLC expression is mainly in the meristem, VIN3 expression is mainly in the meristem. But if you express them elsewhere, you can basically say, you know, all the cells are receiving the temperature. It's just where they're expressed is then their functional place.
Okay, that's very interesting. Thank you.
You know, I talked to a colleague and he was saying, if you look at the root, it's almost like a population of people voting. You have a whole set of people, they're all given a card, they're voting green or red, and they've got to decide, you know, how much cold, because you remember it's not an easy decision. Have I had winter? I need to have got the whole of winter, because if I flower too soon, and I didn't show you this, you know, it's of course terrible from a fitness point of view, because then snow happens as the plants are flowering. You know, the seed formation is really poor. So plants need to judge winter very accurately. And the other thing I point to is that, you know, if you look at cherry trees, they all flower pretty much the same time. They make a very, very measured decision of when to flower. And it's pretty much the same calculation in all plants, in all trees of the same variety. So this population of individual people will have a green and a red, and they're all trying to decide, have I had enough cold yet? And so they then put their card down, and that's nucleation. So they're red. That person has cast the vote. And I think it just takes a long time for that population to all cast the vote. And that's why it's a quantitative effect. That's sort of how I see it, as a population decision. But of course, it's mainly in the meristems that it's the important cells that, you know, it's not every cell in the plant, but you can sort of see it as a decision made by many inputs.
Does anyone have any other questions? If not, I guess I can.
There's one in the chat here, actually, Aaron.
Yeah, should I maybe read it out so everyone can listen to it? So Fiona says, unsure if I got this correct, but am I right in thinking transcriptional repression is induced by cold air once the temperature drops below 0, but epigenetic memory is done by a long-term temperature average? Yeah. Is it possible that at a constant temperature of, for example, below 4 degrees, for epigenetic memory, repression to be induced without transcriptional repression?
Yeah, so basically it's all about these two cold steps that I've described to you. It's cool air, actually, rather than cold air. There is a different one called cold air. So this antisense transcript, you know, I've shown you it's highly stimulated by freezing, but it's also induced by 4 degrees constant. So it's not just freezing, it's just amplified at freezing. And the long-term average, there are many inputs. Epigenetic memory is basically limited by VIN3. And VIN3 has, of course, these many different temperature inputs, long-term, absence of warmth, current. So all of that is integrated through accumulated VIN3, which then promotes this switch. So it's a very complicated system. This is why we've actually done a lot of modeling. And so if you are interested, please do go onto PubMed and search our primary papers, because I think they will explain in a lot more detail, perhaps the bits I confused you on. But they're two rather different temperature inputs. One is affecting transcription. You need transcription to go off, but the other is epigenetic long-term memory. That's again multiple temperatures, but it's also connected with a transcriptional downregulation. So there is a real multifactorial nature to all of this.
I guess I have a question myself as well. So like how differently does vernalization perhaps differ between like different plants? For example, I guess like cool air, just now you mentioned it was a switch at 0 degrees, but I guess 0 degrees might not be as useful for some plants compared to others.
No, absolutely. I mean, that's the joy, and I didn't get into it, but we've done quite a lot of work on natural variation because, in Norwich, 0, well, let's say in the south of France, the Arabidopsis there are not going to experience the 0, are they? They're going to experience other things. So we've actually gone in and looked at how are plants adapting to these different winters. And it seems to be surprisingly non-coding SNPs within FLC itself that somehow modulate how the chromatin proteins are talking to the locus. So those input pathways, there are many input pathways. So, if you don't freeze, well, freezing is not going to help, but one of the other many inputs will then be the predominant factor. But where the natural variation seems to map is then as those factors come in and associate and dissociate from the chromatin, those SNPs affect that association constant. And so you need more or less of whatever that factor is to actually get the switching mechanism. So evolution has just tweaked the mechanism by affecting binding constants, I think. But I would ask, actually, you guys can help me. So I try quite hard to think how to explain this in a sort of an analogy, which is something more people are more familiar with. So you've got a switch and you have all these different inputs, which have, different parts of temperature, but potentially other things too. And as you say, in some places it's not freezing. So now of the 10 factors D and E will be more important for that switch. But up in the north of Sweden, well, it'll be freezing, factor A and B will be more important. But as long as that switch happens, it doesn't actually matter which of those many factors are there. It's all converging on the same switching mechanism, which is the nucleation. So can you come up with a wonderful analogy for me that is a sort of, would help people understand that multifactorial nature, that you can push through many different ways and make one switch? Because it would help me explain this a lot to people broadly. And I haven't come up with a good example yet. So, prize for whoever emails me with the best one. I'll come up with some wonderful reward for you.
Could it be maybe like if there's a lot of taps and water flows through those taps, but at different rates, and once you overflow, and then perhaps something happens.
Okay, well, I can think through that one. Yeah.
We also have a question from the YouTube live stream actually. So this person is asking whether the flowering mechanism which you mentioned is the same or similar for perennial plants.
Very good question. Absolutely. Yes, so I've really focused on Arabidopsis thaliana, which is an annual, but there are relatives of Arabidopsis which are perennials, and basically not all the plant flowers. So there's always a bit left for the next year's flowering, and so it keeps basically going year after year. And a colleague of mine who used to work here, George Krupplen, went to work in Germany, and he tackled that question. So what's the basis of perenniality? And the first mutant he got, where he did a genetic screen to look for now loss of perenniality, turned out to involve FLC. So there's the same kind of mechanism, but in perennials, there's a second pathway involving age, which involves microRNAs and another set of repressors for flowering, so that you've got it's like a double lock. And so once cells are old enough, the age pathway goes away. Now FLC suppression gives you flowering, but the first year the age pathway suppresses flowering. So it's a very nice example of evolution of this basic mechanism into now perennial flowering time. So that's all published, and some nice reviews on that out there too. And I'm a big fan of, if you really understand the mechanism, then looking at evolutionary, trajectories becomes really interesting, because you can sort of see what's tweaked. Evolution just tweaks what's there. And once you get to the mechanistic detail, it becomes a fun project.
Does anyone have any other questions?
How about questions about, choosing a project? Because people always say to me, how did you pick vernalization? But, I want to hear what you guys have to ask.
I think many of us wouldn't mind hearing about that.
Well, because I've worked on this project for 30 years. And, when we started, no one had worked on Arabidopsis before. So it was, first of all, gene cloning, and then we got the genes, and then we had the biochemistry, and then on and on. And it's been a fantastic sort of progression of understanding that people look back and think, crikey, that was a great question. How did you figure out to ask that? And it was pure luck to a certain extent. But, I would say, if you look around your system, and I think plants are a great system because they are so plastic with their environment. It was just, when I lived in California, it was sort of very striking to me that, there, of course, winter's really not very long. And yet when spring came, everything flowered synchronously again. So what were they monitoring? And I went into the shop one day to buy some tulip bulbs. And the man in the shop told me to put them in the fridge for 6 weeks, because sowing them in the ground in California doesn't give them enough cold. And that, I was like, wow, they actually need the cold. And I went away and, read why they need the cold. And that's what got me into vernalization. So, just follow the sort of something that you just think is really interesting. And I think that sustains you for a long time. And I was incredibly lucky, polycomb, so it's nicely conserved, which makes it easier to get funding and so on. And now, of course, this temperature, hugely important to understand how plants are monitoring temperature, especially fluctuating temperature. So that's also been quite important. Oh, here's a question from Shreyas. So Shreyas, thank you for organizing all this. And then I hope your interview went well. Oh, there, Shreyas. Is that how you say?
Hi, Caroline. Yeah, that is.
Have you ever been tempted to switch to a completely different question, and what persuaded you to maintain your focus? I've never really been considered. Because actually, as you dig and dig and dig, it's like doing a very complex jigsaw puzzle, and you can sort of see the outside, but you can't quite see the middle. So I just want to keep doing the middle, So I've never really been, somebody who said, okay, I've had enough of vernalization, I'll do something else. So it's just kept me going. But I am the tortoise in the tortoise and the hare. So, just keep plodding along.
Thanks. Could you comment a bit more on like the evolutionary sort of trajectories which led to the mechanisms which you described today? Do we know about them?
Well, so when we've gone in and looked at Arabidopsis that is adapted to northern Sweden versus southern France, for example. And I think the expectation was that it was going to be lots of small changes in many factors delivering that adaptation. Actually, the genetics, the QTLs and everything kept taking us back to FLC again. And in fact, it's non-coding SNPs within FLC which somehow affect the association of these chromatin regulators to affect the stability of the switch, and also somehow the spreading in this state following, transfer back into the spring and the summer. So there are a couple of papers on that. In one of my talks, the last review that we did, and actually this is an alumnus of yours, Charlie Whitaker, was a Plantsky, is that what you call them, at Cambridge? He came to my lab about 4 years ago and really got excited about Poulet and chromatin silencing, but discovered that he didn't like doing PCR. And actually a lot of molecular biology is a lot of qPCR. So after a year, he went off and did epidemiology. But he wrote a review before he left, which is free, available on the website. And he did a great job at summarizing everything, including the evolutionary questions. So, I point you to that. And actually, we were asked to write it with a title that said, FLC, a paradigm for mechanism and evolution, because people in the evolutionary field, we're not expecting sort of a small number of major changes. And so it was so unexpected that it was slightly ignored for a long time. And so the editors, when they asked us to write that review, asked us specifically to have a title and be a bit provocative. And so Charlie wrote it in this Game of Thrones theme, which I didn't understand because I've never seen Game of Thrones, but I would point you to it. It's an entertaining read, and it's got all of the fairly recent data in it.
That sounds very exciting. Certainly will.
Okay. Okay, well, look, lovely to see everybody, the ones that have put their screens on. I wish you the best of luck. I'm happy to come, as I say, in the summer when things are a little less COVID again, and come down to the pub if people want to talk about stuff just informally. I am in Cambridge most of the time, once a week, because I come to the LNB once a week. So, do keep in touch, and the best of luck.
Thank you for giving the talk.
My pleasure. Okay.
Thank you so much.
Bye-bye.
See you.
Thank you.
Thank you.